Dr. Collison and Dr. Gilbert, how affordable is genetic and genomic sequencing, and what is the attitude of the insurance companies to cover these costs? I think we're getting a bunch of questions on the cost of precision medicine right now.
So if the technologies continue to change, like Dr. Collison said, to sequence the first human genome was more than $1 billion. The last estimates that I've heard for sequencing all of the genes in your genome currently is roughly $1,000, a little less than $1,000 maybe, and it's anticipated that should continue to drop over the next 2 or 3 years. I think a good goal would be, closer to $100. We don't have to sequence all of the genome, so we can save costs by sequencing a part of the genome that we know is highly correlated with disease. So in certain cancers, we know that, say, 95% of patients have these 100 mutations, and we can just sequence those 100 genes and gather most of the information we need. And then, like I said, there are even newer technologies that don't use sequencing, but instead use other enzymes and other newer methods to get at part of this information.
Yeah, and I'll say, just because I think the listener asked for dollar amounts, U.S. dollar amounts, to do a germline test, what genes were you born with? The internet will sell you that for around $100, and those tests are not horrible. They're not medical grade per se. The medical grade versions of those with all the insurance and stuff often goes up to around $1,000. The cancer tests we're talking about with lots of genes is usually closer to $2,000 to $5,000. And the other part of the question was, what does insurance pay for that? And the answer is it depends. And it really depends on whether the disease, lung cancer often reimbursed, colon cancer sometimes. It's really adjudicated on the insurance level by what disease the testing's being done in, because a lot of our reimbursement's still built on, the lung cancer module versus the colon cancer module, whereas I think a lot of us oncologists think more about mutations than diseases of origin, but we try to do both.
I think the related answer to this sort of vein of questions is how much. So diagnosis is one part, but how much do the therapies cost for precision medicine? And that in some ways is a discussion between the drug makers and the individual countries that they're being applied to. And so you see discrepancies in cost between countries that relate to the negotiation between, for-profit companies and national governments. There's a subtlety to this as well that, in my opinion, science sort of is designed to show you what could be done, and then it's up to engineers and production and other means to scale this to be able to treat like a global population. And so in some of the things that we're talking about today, like cellular therapies, are currently extraordinarily expensive. And my hope is that as they're proven to be useful and as they're scaled up, we'll find clever ways to decrease the costs. But that remains to be seen.
Right.
So we have Adam with us now. Adam Hayden is a brain tumor advocate and survivor, and he is actually, his personal blog called Glioblastology is a popular peer-to-peer resource and is in syndication with the Cancer Health magazine. Adam, please ask your questions.
Terrific. Thanks so much. And I was speaking into the abyss, so it's nice to hear your voice, Priya. And Heidi, nice to share the panel with you. I'm going to just, play off of what we were just discussing around cost. So the cost of therapeutic and CAR T cell therapy, et cetera, we know it's extraordinarily expensive, and we've had some good news that CMS is going to cover some of that, so hopefully we'll move in that direction. But I want to ask actually the flip of this. So last year, Siddhartha Mukherjee wrote a cool piece for New York Times Magazine saying that you can compare two blood-thinning medications after heart attack. One costs $6 a pill, the other is $0.25 a pill. He said that things like using the tools of genomics might help us prescribe cheaper medication when it will be as effective in some patient populations as opposed to others. So that was a call for cost savings through what I take to be personalized medicine. So I'd love you just to remark on, is there an opportunity for savings in the long run around this practice of medicine?
I'll comment on that. This is Eric. I think yes. You're talking about efficacy, and that's certainly appropriate. Does this drug work better than the other drug? I would add even a more obvious and first baby step layer. In medical oncology, what I do is I talk to a patient about their treatment. We're going to do X. We're going to put our head down, walk uphill in the snow for 2 months, and then we're going to do another set of scans and see if the tumor shrinks. And if it does, then we'll keep doing that. And if it doesn't, we'll do something else. And so that's fine, but that takes 2 months, and these things are charged by the dose. That's often 4 to 8 doses of this medicine at $2,000 to $5,000 a pop, and these things add up. What if we could tell after 1 dose with a blood draw rather than waiting 2 months? So I think that's an emergent area where actually precision diagnostics might prevent expensive, ineffective therapy much more quickly than the slower diagnostics we use today, which are insurance reimbursed. So paradoxically, it may be cost effective to pay more for diagnostics to save less on drugs, to save more on drugs. And then the other example I'll let Luke comment on. I think he's found many times that a cheap drug actually in a given genetic system works better than an expensive one, and that bias is really in the system here.
Yeah, I think part of the problem is that the way drugs are approved is based off of their activity across a diverse set of patients. So you need to show efficacy in a given disease, or at least within a given disease subtype, which often I think obscures exceptional responders and exceptional non-responders who don't benefit at all from a given drug. And so a lot of my research is around trying to explore this using models, because it's very difficult to design. Eric will have to correct me here, but we don't usually design personalized clinical trials at a single person level, or I don't know how you would design that. And so this is an ongoing science and medicine problem that has sort of legal FDA ramifications too. How do you prove something is effective and safe for an individual versus a population of patients? Yeah,
and there's been some really exciting approaches to that from some of our colleagues in the UC system. The UC San Diego group, for example, had published an interesting paper using given one patient, Susie Q down in San Diego. She got first treatment X, and then she got treatment Y, and it's the same person. And if she was on treatment X longer than treatment Y, then maybe it's fair to compare treatment X and Y with her genome and say this is kind of a clinical trial of one person, but done sequentially rather than simultaneously. There's caveats to that, right? The tumor's grown in that ensuing time, and it may not be a fair comparison. But I think we're learning ways to do these single patient and personalized trials as we go.
Okay, terrific. So here's within the world of brain tumor and brain cancer advocacy, what we see sometimes is that our newly diagnosed patients or not yet diagnosed patients will show up at the emergency department with some emergent symptom, seizure or something, and oftentimes are sort of admitted and whisked into the standard of care protocol quickly, making them ineligible for newly diagnosed clinical trials or unable to spend the time on the diagnostic screening that you're addressing. So I'm curious, something we think a lot about in the advocacy community is, how do we help folks that are not yet even diagnosed understand options before pursuing with a standard of care protocol when something more precise may be appropriate or available? Could you remark on, just in the settings of clinical care, how do we better inform people to make these choices?
Yeah, it's a huge problem. We have the same thing in pancreas cancer, this late presentation problem. My colleague Brian Wolpin at Dana-Farber presented some really compelling data teaching a robot how to look at CT scans through the whole Partners Health system, and it was really exciting that they could actually find, some cases of pancreas cancer before just on the imaging that they had done for other reasons. And so where I think this puck is going is that we're going to have systems working in the background while the radiologists are still going to have a job. There are going to be, computers looking at the brains of these people and setting off alarms when there's a tumor seen, hopefully to the glioblastoma network. And that will trigger, let's get the consent team down there in real time, kind of on the equivalent of the 2019 Batphone, to consent them in real time. I can't think of another way of doing it because we just can't post an advocate in every emergency room 24/7.
That's exciting work, and I love the Batphone.
Yeah.
I mean, just picking up the glioblastoma Batphone. I'm not sure that's good or bad. That's terrific. One other thing that actually just came up during the discussion, and we talked a little bit about the KRAS case earlier. I'm curious. Do you think that are we moving already in the direction of treating the molecular profiles rather than the tissues of origin when trying to better understand treatment plans? It seems like we had some conversations, so I'd just like to hear more about that.
So I think that idea was popularized, that we can move away from a tissue-based understanding of disease towards just a molecular profile. I sort of disagree with this idea. I don't think it's been borne out. In some cases it has, in many cases it hasn't. And so I think that we need to consider the molecular profile of a given tumor type and stratify our treatment-based decisions based off that. But I'd love to know what Eric thinks.
Yeah, I agree. I think the bloom is not entirely off the rose on that, but it's not a slam dunk. And glioblastoma is a perfect example where we had drugs to treat some of the common mutations that we saw in lung cancer and glioblastoma, but it just turns out due to brain penetration and the biologies being different, they just weren't as successful in glioblastoma as they were in lung cancer. And to be honest, we didn't really ever figure out why. We're still working on that. So I think, as with all breakthroughs, it comes with footnotes, and to really know it, you got to read the whole book and get to the bottom of it and keep going. So we're getting there. We do have medicines that are used across cancers. Gastric cancer and breast cancer share mutations. Melanoma and colon cancer have FDA-approved drugs that are the same. But how well they work and how they're combined with other drugs differs.
Great, terrific.
Thank you. It's kind of time to wrap up now, but I have just one more question because this is exciting. I believe Dr. Collis and Dr. Gilbert, you folks have just recently moved into the new UCSF Center, which is exclusively for precision medicine. So it would be great if you could let us know what the new center offers in terms of trials, research, etc., and then we can wrap it up.
Well, it's a beautiful building. We moved in it's been a month now, and I've had about 4 or 5 clinics in the new building. I think some of the perks are that every level has its own treatment level there, so you can actually see the patients getting the treatment on the same floor. Often they'll go away and we won't see them again until the next visit, 2 weeks. So that's a nice feature. And also the intermingling of different professions, radiation oncology, surgeons, are much more interspersed than they have been. And I think that's the key to success in these diseases, the multidisciplinary approach.
I would add that I think there's one thing that may be missing here is that I've never set foot in this building. And so UCSF has rich history of intermixing basic researchers who are not MDs with clinician scientists, and so I think that the cancer research building is a wonderful mix of PhD, MD, and MD PhD scientists, And I think I should go over to the new cancer building and say
lunch.
hi.